and then from there, I've got to learn. I just clicked on share a screen, and it's asking me, I guess, which screen I'm supposed to share. Here we go. It's PowerPoint. And yes, I entitled the talk, How to Kill a Neuron. And it sort of wraps up in one sentence the processes that seem to happen that I've just learned about over the last month, that what actually happens, because as you know, with Parkinsonism, you lose neurons, and actually they're a very specific class of neurons, which is also true, by the way, in Alzheimer's disease. Alzheimer's disease, you lose neurons that make acetylcholine, which has to do with storing memories and with alertness and things like that. And in Parkinsonism, those neurons don't seem to be affected very much, but it's the ones that have to do with motor control. So it turns out that they don't have to die, that it's a process that the body carries out, which is probably protective in the case of something like cancer or an infection. It's a good thing to have happen, but in this case, it seems to have gone awry. Anyway, let me see if I can get this started here, and I'll go through the slides. And please stop me anytime that you want to ask questions. There aren't very many people tonight, and it's going to be easy to jump back and forth between these figures and any kind of discussion that we want to have. I'm a little more experienced with this system now than I was last time, so I think I can use this little red pointer. I can't make a permanent line on the slides, but I can indicate important points with this pointer, a little red dot. Can the rest of you see the little red dot?
Yeah.
Okay, great. And I can still advance the slides, I think. I thought I could. Yes, there we go. Okay, so I'll go back again and be an old-fashioned professor and talk a little bit about the anatomy. Whoops. And then after the anatomy, I'll talk about the fact that a lot of the cells in the brain are not nerve cells. And in fact, they're, well, non-neural elements that support the nerve cells and create the environment where the neuron lives. And I want to come back to this idea about the prion diseases and the alpha-synuclein that we talked about last month. But then we need to talk about immunity and just how the body fights off infection and how it works in the brain. And finally, towards the end, we get to the gut-brain connection. And remarkable information that I don't know how to interpret for you will end up with, and that's a scientific paper that was published on December 2nd. So it's hot off the press, and well, we'll decide what to do with it when we get there. So, as I just said in the last slide, brain is composed of cells, but less than 50% are neurons. And I forget this too, because I spent my life studying neurons, and most of the brain is supporting cells. And they support and they nurture the neurons, and they're generally called neuroglia. So neuroglia the brain consists of neurons and these neuroglial cells. There's four major types: the ependymal cells, the astrocytes, the microglia, and the oligodendrocytes. And it's really the astrocytes and the microglia that are what we'll talk about today. But first, just to go back to this idea that all these supporting cells really protect the brain and they enclose it. The brain is inside of the brain case, inside of the bone, and inside the spinal cord is inside of the spine is the spinal cord. And this blue color that you see here. Oh, by the way, Peter, if you want to share the slides with everyone, I don't know if you normally do that, but I sent them to you, and you're quite welcome to forward these on to anybody who'd like to have a copy. And my address email is there someplace, so I'd be happy to answer any questions. So after the talk, you can send the actual PowerPoint off to people if you'd like.
I'm happy to do that, but the PowerPoint that you're showing with your explanation is more informative. So
that's fine. I just if anybody wants it, they can have the dry images as well.
Sure.
So anyway, the brain there is floating inside. This blue stuff is supposed to be the liquid, the watery liquid that supports the brain. It actually floats inside of the brain case, inside of this fluid. And the fluid's made by one of the supporting cells called the ependymma, or the ependymal cells. I'll come back to that in a minute. The neuron itself, we see this characteristic cell with all these little branches sticking out where the synapses occur, but we never talk about what's in between, which in between all these spaces, in between the branches of the neurons, are dozens of little cells that stick to it and provide nutrient and mechanical support. And another special class of nerves of supporting cells covers the axon. The axon's running down here. It's that little brown line in the middle. And then there's the oligodendrocytes, a supporting cell that wraps around and around and around and around the axon of the nerve cell and insulates it, literally electrically. It's an electrical insulation. So that, in fact, and what it does is it helps the electrical signal travel because it's insulated. It can travel farther and it doesn't short circuit, essentially. And so this is a fatty material that goes around the axon. It's a special cell that makes that. So I've talked already now about the ependymal cells that separate the brain from the rest of the circulatory system and generate that fluid that I was talking about. And I also talked about the oligodendroglia, which make that insulation that insulates the nerves themselves. So now I'll talk about astrocytes and microglia. But in general, these supporting cells surround the neurons, hold them in place, supply nutrients, insulate them from one another. And the key thing for us tonight is that these cells also are responsible for destroying the pathogens and for removing sick or dead neurons. So, well, I can probably skip over this. This is the ependymal cells again. They line the surfaces around the brain. They keep the environment rather constant because they actually filter things between the blood and this fluid around the brain. And they take out things that are not necessary and they add things that they selectively bring in things that the brain needs. And then once the fluid has circulated for a while, it goes back out into the venous circulation in the midline of the brain, actually. Okay, so the next category is the astrocytes. They're roughly 30% of the brain cells. And if we have this little bag that we see here, this little kind of box with rounded corners, we'll call it a neuron. You can see there's all kinds of chemical reactions inside of the neuron. And many of those processes that are necessary to keep the neuron alive are assisted by the astrocyte. The astrocyte seems to be sort of a caretaker and supporting cell that trades various molecules. And I don't see the fatty acids or the lipids that we were talking about earlier tonight, but they must be in there someplace. They're passed back and forth. And various amino acids are passed back and forth between the astrocyte and the neuron. So they have a very intimate life together. The other class of cells that I mentioned is the oligodendrocytes, and they extend their little processes and insulate the axons. They don't pay much attention to the neuron cell body, but they put insulation around the axons and allow the electrical signals to travel more effectively. And so someone with multiple sclerosis, for example, it's becoming apparent now that multiple sclerosis is a kind of an autoimmune disease where your body builds up a reaction and tries to treat this insulation around the nerves as a foreign material and attacks it and tries to digest it. And as you know, in multiple sclerosis, there are periods when the individual will be weak, almost paralyzed, and then they'll recover, and then they have another attack. And each one of those attacks is white blood cells and the microglia attacking the insulation around the nerves. And so that's this particular cell, the oligodendrocyte, is important in multiple sclerosis, but not so important for us today. One of the tissues that is important is the microglia. The microglia are tiny little cells that lie in the spaces in between the neurons, and they're usually quiescent. They count for 10 to 15% of the cells in the brain. But if there's, in this case, is traumatic brain injury, where there's a blow to the head, they become activated by perhaps the injury of the tissue around them. They get bigger in size, and they begin to actively look for damaged tissue that they can consume and phagocytose. They eat it, actually. They incorporate it within their cell body, and then they carry it away to be taken out through the circulation. And they are the main form of immune defense. If there's bacteria in your brain, the microglia will come along and pick up the bacteria and carry them away. So they're necessary. There's essentially the white blood cells. The same role that a white blood cell will play in an infection in your skin, these cells play in the nervous system.
Viruses?
Viruses also, they can inflame the brain. If the virus actually kills a cell, then these cells come along and pick up the bits and pieces. And so, yes, they do. If they detect that a cell is infected with a virus, then they will actually engulf the cell even before it's dead. So that way they capture the virus and keep it from spreading.
That's good.
Yeah, so that's good. Now, but then later on this evening, we'll see that there maybe are times when we wish they weren't quite so effective. So now so that's a little bit of anatomy, and that's what those cells do in the brain. So now let me talk about immune response, by the reaction to infection for the rest of the body. Anytime the body has a disease process going on, the same kinds of things happen. There are cells, the white blood cells, that come along and pick up the bacteria, or if a cell is infected by a virus, they'll engulf the cell. The way they know to come along and do that is there are chemical signals. And these chemical messages that are released by this cell that is sick or released by the bacteria, somehow they're recognized as being foreign by the white blood cell. And the white blood cell then goes to that place where the highest concentration of that chemical is and begins to digest the tissue in the environment. And here's the same story in pictures. This line down here, this is supposed to be a tube, either a lymph vessel or a red or a blood vessel. And in an area where there is an infection, and here's the bacteria right here, the white blood cells and monocytes come out of the circulation. They actually crawl into the tissue and begin to engulf the bacteria. And I included this picture because it has all kinds of abbreviations. There's LTB something or other, I can't read without my glasses, and IL, and LT stands for leukotriene, IL stands for interleukin, And there's TGF, I've forgotten what that is, but these are chemicals that are made in a region of an infection, and they're the chemicals that attract the white blood cells. And up above on the top here is when cells die, when the tissue is so sick that it dies, it releases arachidonic acid, and that turns into, with an enzyme called COX, it breaks down into prostaglandins and leukotrienes and makes those signals that bring in the white blood cells. And so there are all kinds of chemical signals that we don't need to worry about tonight, but they trigger the defense, the inflammation in the tissue. And then as the disease is engulfed by the white blood cells, as the bacteria are digested and taken away, then some of these same molecules begin to signal that inflammation is not necessary anymore, and the tissue begins to become less red and less inflamed. But again, it's the interleukins, and this is, I think, another leukotriene here. Other molecules that are similar to those that signal inflammation, they signal that it's now time to calm down and go back to business as it was before. And gradually the white blood cells carry the infected tissue away through the lymphatic vessels. So that's, in 2 minutes, that's kind of the response of the body to a bacterial infection. So it's these chemical signals from these molecules that you may or may not have heard of before, the leukotrienes and the prostaglandins, but also these same or similar molecules are then involved in ending the inflammation. That's important. We'll come back to that. So now if we go back to the brain. In the brain, it's not white blood cells, but it's the astrocytes and the microglia. But they work the same way. They communicate by chemical messengers that float around in between the cells. And if there's a lot of leukotrienes and so on, then the astrocytes and microglia become alerted to the fact that there's a foreign substance in the area. And they turn essentially into white blood cells, or they actually send other signals that call white blood cells to come into the brain. The main signaling cell is the astrocyte, and it receives all kinds of chemical signals, and it integrates those and gives off signals that can bring other cells to help control the inflammatory process. And here's this poor neuron sitting down here in the corner, and the astrocyte actually is kind of leading the battle, giving instructions about whether white blood cells should come, what the macrophage is supposed to do. And again, it's the same molecules. It's the IL, the interleukins that are important. These things, I don't remember what they are, but you could pick up a reference, or I can send it to you if you want to read about how this works. So this astrocyte then is central, very important. And then the instructions of the astrocyte are carried out by the microglia. They become the scavengers that clean up the brain. If you wish, they're the soldiers of the central nervous system. And they follow the instructions either from astrocytes or white blood cells. If the astrocyte calls in white blood cells, the white blood cells can give off chemicals that will encourage the microglia to engulf and digest things in its environment. So you can think of them as soldiers, and the astrocytes give their orders, and the astrocytes release the interleukins, the prostaglandins, the cytokines, and also they complement, that's antibodies. So then the microglia, they must recognize a foreign, once they get these signals to go looking for something that's foreign, they have to figure out what it is. And the way they do is that usually there's specific molecules on the surface of the cell they're supposed to swallow and digest. So they have to recognize them as foreign. They engulf them, and they too work. They can call a specific kind of white blood cell called the T cell to help them eat the foreign material. And once it's kind of a cascade, and once it starts, it's like a feeding frenzy among sharks. More and more cells are called into the area, and the local tissue is digested and carried away. So here we are then with the trying to get this little thing out of the way. So essentially, the astrocytes are the intimate partners of neurons, and they sense the neuronal health as well as the health of the other cells in the environment. So they play a very important part in the repair of the nervous system, because they essentially lead the orchestra or lead the battle. They're the cells that give out the commands about digestion and cleaning up. So now I've done something. Yeah, here we go. Yes. So just to summarize, I've told you an awful lot of material in a very short amount of time. So let me see if I can summarize it here. The astrocytes detect foreign substances. They do play a role in repair. And if there is actual physical damage in the brain, like a stroke or like a mechanical wound or a cutting of the spinal cord or something, then they'll produce a scar. But they play a leadership role in controlling any inflammatory process, and they send out the signals that set the stage for the microglia and the white blood cells to come and clean up the area. By contrast, the microglia essentially follow instructions. They actively destroy and clean up the debris, and those are the cells that we find in and around the Lewy bodies, and we find them around dying neurons in the substantia. Sorry, I was going to say substantia gelatinosa and the substantia nigra. As I said, they follow instructions from the astrocytes. So, in summary, the astrocytes are constantly monitoring their environment for signs of abnormality and disease. When they detect substances from other cells, from neurons, or from the blood, they then produce chemicals that sensitize and cause this reaction, this inflammatory reaction I was just talking about. What did I do? Did I go backwards? I think I duplicated a slide. I'm sorry. We'll skip over that. Okay. So now I want to add something, and there's a very interesting review article by this man Rothammer, and I'd be happy to send it along if anybody wants to read it. But this is a picture from his paper where he talks about the processes that I've just talked about, where there's substances in the environment of the astrocyte that encourage inflammation and encourage the astrocyte to start the digestive process. And there's interleukin 1B, interleukin 6, NO is nitrous oxide. I can't remember what the CCL molecule is. And these are the what did I call them? Cytokines. These are cytokines, but very similar molecules like this cytokine or this interleukin 10. I believe this is another cytokine, but I'm not sure. And tell the astrocyte that it's time to calm down. And these molecules are anti-inflammatory. They have a calming effect upon the nervous system and the cells that might digest other neurons. So let me just jump back now to what I talked about last month. And this is the alpha-synuclein that, remember, is a very important molecule, and it's one that seems to be intimately or centrally involved in Parkinsonism, in the sense that it is perhaps produced in excess, but even if it's not, it's certainly misfolded, and it tends to accumulate. Once it's been misfolded, it's hard to digest, and it accumulates in the neurons in Parkinsonism. And you remember last month I said that it's particularly located in the energy producing units of the cell, the mitochondrion. And obviously that's a really important part of the cell. And alpha-synuclein is located there for what reasons exactly, I don't know. The other place is that it's involved in carrying materials from the cell body down to the synapse. And of course, the synapse can't maintain itself if it doesn't continue to get nutrients from the cell body. So then what happens in Parkinsonism is that when these synuclein proteins are misfolded, they form tangles, and they actually form extracellular clumps called Lewy bodies, and they may accumulate and distort the shape of the synapse, so probably the synapse does not work well. So when neurons contain misfolded alpha-nuclein, do the astrocytes detect this? Are they able to recognize that there's a change inside of the neuron that is not healthy, in other words? If so, could they tell the microglia to eat that neuron? And so this is the process probably whereby the body, your own body, tells itself to digest its own neurons because they're detected as being unusual, being not as healthy as the neighbor, and that's as a signal that in evolution has protected us from cancer and from other diseases. But in this case, perhaps a functional cell is being destroyed by the microglia in the brain. So that's the question. Can the abnormal alpha-nuclein make a neuron sufficiently sick or unhealthy that the rest of the brain detects it as being unhealthy and then starts this inflammatory process? And the astrocyte, which used to be the supporting element and was anti-inflammatory, is converted over to the other side where it's pro-inflammatory and begins to give off the signals that the microglia then use to say, okay, it's time for me to start looking for something to eat. And I leave that with you. That's the state of research today. You see this paper, Rothammer, was published in 2016. This is just, that's a question that is in the field today. What this idea is, what's interesting for me at least about this idea is, if this were true, could we influence that balance? Can we push the astrocyte back towards the anti-inflammatory side? And can we make them the astrocytes less responsive to inflammatory signals? Can we block the activation of the microglia? Can we reduce the number of chemicals in the brain that promote inflammation? And it turns out there's a very specific receptor called the fractalin receptor on microglia that really sets it off. So maybe there's some drug or something that might block that receptor. And I'll talk about the germ-free mice in just a minute. So that's where I was going to leave the story, essentially, a week ago. I was going to go and start talking about diet and how maybe diet could influence the chemicals that get into the brain and so on. But instead, as I said, this paper was just published a few days ago, and I want to end. I want to end with another bit of information about how we might influence that balance. Before I go there, I need to talk about the digestive system. So here's the the thing that has become very popular in the layman's literature in the last year or so. There is a very clear connection, and when I was in my doing my research, and when I was at McGill or the University of Montreal, if someone had told me what I'm telling you today, I would not have believed it. And yet, in the last couple of years, it's become quite clear. There's a lot of scientific evidence for a very clear connection between diet, microbiome, and the brain. And the microbiome, I'll define in the next slide. That's all the